Hello, my name is TJ Flynn, and I go to the Wilmington High School, and this is my science fair project. My science fair project was on how different substances affect the growth and development of yeast cells. A yeast is characterized under a category called Saccharomyces, which means sugar eaters. Yeast really love sugar, and they need sugar basically so they can reproduce and make more yeast. 3 ways of reproducing yeast is budding, binary fission, and reproduction. In the process of budding, a yeast will grow what's called a bud on the cell wall of that yeast, and soon that bud will grow into new yeast, and that yeast will have a bud, and it'll just keep budding and budding and budding. The second way is through binary fission. And this simply happens when a yeast splits into 2 different yeast cells. And the third way is through reproduction. This happens when 2 yeasts meet together and they reproduce about 6 to 8 spores, and those spores will soon turn into yeast. This is a picture of budding yeast, that's a picture of binary fission, and that's a picture of reproduction. There are 2 ways that yeasts get their energy to do this reproduction. The first way I will talk about is cellular respiration. In cellular respiration, the yeast cell simply breathes in oxygen when it's available. And usually the leftover parts are water and carbon dioxide gas. The second way is through fermentation. The yeast will use the fermentation process when there is not enough oxygen. And what happens is the yeast has little ferments inside, and these ferments will break down the sugar. And the leftover parts are ethyl alcohol and carbon dioxide. And fermentation is usually used in the making of alcoholic products. Why I did this? Well, one day when we were in biology class with Mr. Fadi, we were talking about yeasts, and the thought came across my mind that I want to find a way that I can control the growth of yeast. So that's when I decided on my title and decided on my project. These are some of the tools that I used for my project. These are some test tubes holding some of the growing yeast. I used a notebook to write down all my findings. I used a hemocytometer, which is, it has little grids on it, and what you do is you have to dilute the yeast, and when you dilute the yeast, you count the number of them and times it by 50, which is the dilution factor. And the way you dilute the yeast is with the pipette. What I did was I drew the yeast up to about the 0.5 part, and then I drew in water all the way up to the 11, and mixed it around and put some on the hemocytometer and viewed it under the microscope. Now my procedure is a little complicated. What I did was I filled this beaker right here up to about 350, and then I filled molasses in about 25 more millimeters, and I used molasses because molasses has a high sugar content and yeast like sugar. And then after that I put in about 1 teaspoon of yeast, and I let it grow for about 10 minutes. And after 10 minutes, I used this formula. Well, actually, after 6 hours, I used this formula. But for the 10-minute part, I put in my findings of how many yeast I found. And the first one was 14,300 yeasts per millimeter squared after 10 minutes on my first experiment. And on the second one, after the 6-hour experiment, it was 26,300. And then what I did was I used this formula here, the 10 minutes over the 10 minutes over the 6 hours, and the question mark over 100. And the question mark was the growth rate. And when I came out, after multiplying, cross-multiplying, I came out to about 83.91% growth rate. And that's going to be known as the known growth rate. Now, on my second experiment, I decided that I want to increase the growth. So what I used was orange juice. And I put in the 350 mLs of water, the 25 mLs of molasses, and about 20 mLs of orange juice. And I figured they're going to grow a lot because orange juice has a high sugar content. And after the 10 minutes, came out with 13,100 yeast per millimeter squared. And after the 6 hours, I came out with 33,800 yeast squared. And with a growth rate of 158.2% growth rate. And I thought that was good. I thought I made a good hypothesis, and it came out. And for the second experiment, the third experiment, what I did was I used milk because I wanted to retard the growth a little bit. So I put in 20 mLs of milk, and after 10 minutes, I came out with 13,200 yeast per millimeter squared. After 6 hours, 17,600 yeast per millimeter squared. And I came out with about a 33.25% growth rate. And that was good again because my hypothesis was to make a low growth rate. And I figured after that if I made a low and a high one, I'm going to try to go for a middle one. So I guessed at about 75 to 80% growth rate. And what I did was I used jelly because I looked at the back label of the orange juice and I found out it was about 25 grams. And I looked at the milk and I couldn't find any grams of sugar. And then I looked at the jelly and it was about 15 grams. So I figured that was about in the middle. So after the 10 minutes, I had 13,000 yeast per millimeter squared. And after the 6 hours, I had 20,855 yeast per millimeter squared with a growth rate of 60.52%. And I was close, but I wasn't close enough to say that my hypothesis was correct. And that's my project. Thank you.
Breslin, I'm a freshman at Wilmington High School, and I did my project on artificial subjectivity of stimuli to the internal clock. It's just about the biological clock within us. I had 3 experiments, and the first one I did was I took a known plant to have the biological clock in it, the oxalis, and I took it and I subjected it to artificial light, to normal sunlight, and to total darkness for a period of 72 hours. And what I found was that after a while, it's supposed to open in the morning and close at night. And what happened was, for the first 24 hours, everything went fine in the artificial light and the darkness. But after 48 and 72 hours, I found that the leaves started opening and closing on their own at different times than they should. So what I conclude from this is that artificial light and darkness have some effect on the biological clock. And the second experiment I did was I tried to reset. I wanted to see how long it would take to reset the clock after I had subjected it to artificial light. And that's I had these pictures here. These are the different. Right here is the box I used for the artificial light, so it would get dim light and not bright light. And here is the normal plant, and it's at a closed position. And this is also at a closed position. What happened was it took about 24 hours for the plants to reset to their normal cycles. So that's what happened there. The last experiment I did was I took my temperature during different times of the day because it is known that temperature has the biological clock has some effect over our temperature rhythms. So what I did was I took my temperature at different times of the day. I took it at 6 o'clock a.m., 1 o'clock p.m., 6 o'clock p.m., and 10 o'clock p.m. And I tried to find out what was the average temperature at those times. And what I found was it was about 97.5 at 6 a.m. That's the lowest, and the highest was at about... The highest was about noon, and that was about 98.7. And what I found out from this is I put myself under different conditions to see if it would have some effect. But the second day, at these times I took a cold shower, and after I saw if it had any degree difference, and it didn't. It just had a teeny bit. It didn't really have effect on the rhythms, so I concurred that it does. The peaks in temperature are caused by an internal clock within. Let's see what else. I have some facts about the internal clock. Most babies are born between 1 a.m. and 6 a.m., and this is because pulses are sent from the mother to the baby, which allow this to happen. The most common time of death is 6 a.m. because most heart attacks peak between 6 and 9 a.m. Most Olympic records are set in late afternoon because there's an adrenaline rush in mid-afternoon after the first one at about 6 a.m. And the second one is side effects from medications are more prone to be strong if administered in early morning than late afternoon. This has to do with also asthma. If you have asthma, then you take antihistamines. Then you should take them in the morning, not the night, because histamines are more they're produced better at night, so it would block off the antihistamines. Brain activity peaks between noon and 3 p.m. People feel a dip in activity at 1 p.m. This is because at 1 p.m. there's a big dip in temperature. And at 6 p.m. production of endorphins is let out is low, very low at that point. So I wouldn't suggest going to a dental appointment during the time. And that's about it for that. So what I really found out is there's an internal clock within us, and what it really is it sends out pulses, and these pulses, they keep track of time, and there's many theories on what they think may cause these pulses. One says that the buildup of proteins in protein synthesis may cause it. One says that the cell membrane, when it gets blocked up, that it sends out a pulse. Every so often, when it stops getting blocked up and goes in, this keeps track of time. So they're not really sure, but they think they both might be right. Both of them combined might be what the internal clock is, and that's my project.
Hi, I'm Cathy, a freshman at Wilmington High School, and I did my project on the effects of light intensity on plant life, and I'll tell you a little bit about the experiment that I did in just a second, but I thought I'd tell you something about photosynthesis, and it's the process in which light from the sun comes down and hits plants. The plants then take the light and turn it into food for the plants so that they can grow and produce oxygen for life on Earth. And we see that happening every single day on Earth, and that's how life survives. All right, this is what I did for the experiment. There were basically three steps. First, it involved 4 different plants, and what I did was I took plants or seeds from radish. I took radish seeds, planted those, and planted them in 4 different pots almost. And radish seeds grow pretty fast. That's why I used it would be good seeds for the experiment. So I planted those, and these are the actual pictures of how they came out. And what I did was I took the 4 different plants. I grew one of those plants naturally with light coming. I put it right next to a window, watered it, had the light came through the window, and basically just the way you would grow a natural plant. Another plant I grew under 24-hour artificial light, and what I did was I grew the plant underneath an artificial fluorescent light, which I had beamed down on the plant 24 hours a day. I never took it off. It was there all the time. And then another plant, actually, I'll show you which ones. This one is the one This is the plant that was exposed to the 24-hour artificial light, and it was placed under fluorescent light. Like I said, the results didn't differ much from the plant that was placed under artificial light for only 12 hours at a time. The plant growth here reached about 7.1 centimeters. The plant stems were green, and the overall plant looked pretty healthy considering it was under artificial light all the time. Okay, the plant that I grew in sunlight is right here. This one by far grew the best. It grew 9.1 centimeters, and this looked also pretty healthy. But the plant stems, they were green. They differed from about 9 centimeters. Some were only 2 centimeters. And so this is the plant that was grown naturally. The other two plants I grew, one was in complete darkness, no light whatsoever. And that's this one here. That actually, I was surprised that it grew at all. It grew to about 8.2 centimeters. But the stems were really white, and they appeared really weak and didn't look as though they were doing so well. But I was surprised that they grew at all. This plant here was, I put that under half artificial light and half darkness. I put this plant right here, I put under the fluorescent light for 12 hours a day. The other 12 hours I put the plant in just complete darkness. So it was almost like a day-night effect, but it was artificial. And so that's the results of the experiment. And this right here is actually this. This is the plant that was grown in 24-hour artificial light, and it's right here. What I did was I added worms to the experiment, and I put the worms in here and sealed off. You know, you can actually see some of the dew on the top of the container. And what I did was I sealed it off so that no air could get in. And the plant, the object of that was, with the worms put in there, the plant growing. I wanted to see if the plant can grow enough or can produce enough oxygen so that the worms can survive. And this has been covered for, I'd say, about 2 weeks now. I haven't really checked it yet, but I will. And so the object of this was to see if the plant that I grew was going to be able to make the worms survive.
Makenna, I'm a freshman at Wilmington High School, and this is my science project. It's on, do sound waves affect plant cells? The reason why I came up with this science project idea is because I was watching TV one night, and on Dateline NBC or some show like that, I saw a thing on agriculture in America and California, and it was telling about how earthquakes affect plant growth, and I just figured, since sound waves are acting almost the same way as like earthquake waves, that I wonder if sound waves could affect plant cells either. So these are all the steps I took. In the first step is I bought a plant called a narcus. It's a plant commonly used in fish tanks. Then the second step I did was to take a narcus, and I took 7 strands of a narcus and put them into 4 jars. One control strand. Second group, I subjected it to listen to rock music. The third group, I subjected it to classical music, and the fourth one, I read to it. And let's see, I did each of these for half an hour for 10 days, and I took logs when I was doing it. I wrote down the day, the times, the temperatures, what kind of light was in the room at the time, what group it was, and how loud the sound was. This is a typical anarchist strand, what it looks like basically. And each night I subjected it to each type of sound wave, and after day 3, day 5, day 7, and day 10 was the final day, I took sketches of each group, and these are the sketches of group 1, These are sketches of group 2. And these are sketches of group 3 and group 4. And group 1, on day 3, the control group, this was day 1. And basically all the strands look like that on day 1. On day 3, the first group, the cell walls got rigid, and that's basically the only change there. In day 5, oxygen bubbles started to appear, and the cell walls got a little more rigid. In day 7, the cell walls got even more rigid, and more oxygen bubbles appear, but brown spots started to appear on the leaves. In day 10, half of a leaf was brown, and oxygen bubbles were all around, and cell walls were starting to break up. For group 2, I was subjected to rock music. I let it listen to a 9 Inch Nails CD. Basically on day 3, the cell walls started to spread out, and the cell walls started to spread out and die, basically. On day 5, the cell walls started to spread out even more, and oxygen bubbles started to appear. And day 7 was basically the most change in any group. The cell walls just basically broke apart, and brown spots started to turn up on the leaves. And day 10, not much, basically the same, not much change. Just more brown started to appear, more oxygen bubbles started to appear. The classical music in group 3 had the least amount of change. In day 3, there was basically no change at all. Day 5, it got a little bit rigid. Day 7, it got a little bit more rigid. And day 10, oxygen bubbles started to appear. The group I read to, not much change there on the 3rd day. On the 5th day, oxygen bubbles started to appear, and part of the leaf started to become brown. On day 7, the cell walls became rigid, and more oxygen bubbles started to appear. And a little bit more of the leaves started to become brown. And on day 10, the cell walls became even more rigid, and more oxygen bubbles started to appear, and part of the leaf became even more brown. The control group, I just let it sit in my kitchen for the 10 days. I didn't do anything out of the ordinary to that. Group 2, I subjected it to rock music. I let it listen to a 9 Inch Nails CD. Group 3, I let it listen to classical music. I let it listen to the Monks of Santo Domingo or something like that. And group 4, I read Spot Goes to School to it. Let's see. Yeah, these are my genres: control, rock, and classical, and reading. As for physical changes, so far the control group is basically worn out by now. It's still wearing down. A lot of the leaves are brown now. The rock group is basically dead. It's dead. The leaves are falling off, and part of the leaves, if they're still on, are black. The control group, the classical group, still look healthy. There's not much deterioration going on in there. And group 4, the one I read to, was almost the same as the control group, but there's a little less deterioration. Let's see. Like I said, I did this for a half hour for 10 days straight, and my basic hypothesis was right, that rock music would basically kill the plant, and the classical music would almost help it out. And the reading of the control group, I really wasn't sure what would happen to the group 4, but the control group, I just thought that was just going to go through normal stages, and I wasn't really sure what was going to happen to group 4. But my hypothesis was basically correct. and that's my project.
Freshman at Wilmington High School, and for my science project, I did the effects of an electromagnetic field on plant growth. The first part of this project I think I should discuss is what exactly is an electromagnetic field. I have a definition written down that I'll just recite for a minute. An electromagnetic field is a region which alternating lines of force and electrical lines of force move at right angles to each other and is created by electrons oscillating in an electrical conductor. Put simply, that's basically saying that whenever current passes through a conductor, such as a wire, a temporary field forms around it, conducting both the properties of an electrical field and magnetic field. This is shown over here. This black line represents a wire, and it shows how the electromagnetic field goes in 360-degree radius around it. The electromagnetic field can be increased by forming that wire into a loop, in which the lines of force intersect in the center of the loop. This is also can be more increased by forming a solenoid, which is basically a coil of wire, in which the center, all the wires have lines of intersecting force in the center of each circle. Therefore, the strength of the solenoid is equal to the strength of each of its individual loops. Now, the reason I chose to do this project is that I figured electricity today is so commonly used that maybe we misjudge it a little bit and consider it safer than it really is. I figured something so powerful to power cities and it was basically been the structure of our modern life has to have some sort of side effect to it. Now, I know that there's presently known dangers to extended exposure to electromagnetic fields. The U.S. government is currently investing about a couple $1 million to find out what are some of the side effects of prolonged exposure to electromagnetic fields. Some of these dangers are depression, increased stress, birth defects, childhood leukemia, miscarriages, cancer of the nervous system, brain cancer, breast cancer, Alzheimer's, lymphatic cancer, and learning disorders. So basically, I knew there was presently known dangers, and I wanted to see if there was anything affecting growth. My hypothesis was that the electromagnetic field would affect the growth in some way. I've most likely predicted that this growth would be retarded, since that the electromagnetic field would interrupt with the chemical processes involved in plant growth. To do my experiment, I built this apparatus, which is basically made out of wood, and it has a standard AC current running through these wires, which form as close as I could to a circle around the plant, which grew inside. The lights is formed as a way of telling that it was working, and to simulate a common household electrical appliance in a better way. The light was covered during the experiment to keep it from interfering with anything else. I ran current through it at all times, and I measured the growth every 2 days. I grew 2 groups, a control and a variable group. Each group consisted of 5 plants. I chose the iris since it grows straight and is relatively quick in growing. I charted the results, and I found that the average growth for the control group after 23 days was 3.5 inches. The average growth for the variable group was 6 4/5 inches. So you can obviously see there's a great difference. Now, I'm not sure if there could have been some other thing that I didn't see, like genetics or something like that may have interfered in the experiment, but I tried to keep the plants as similar as I could. Each plant was the same age, was in similar pots, and had similar soil, and was given the same amount of water over the time. So basically, I came to the conclusion that the growth was affected greatly by the electromagnetic field, even though it appears that the plant inside the field, which is dead now since it's been in my biology class for 2 weeks, but even though the plant's dead now, it shows that the growth was accelerated. But this isn't necessarily a good thing, as we all know that many diseases such as cancer are caused by accelerated cell growth. So basically, electromagnetic fields could still be a threat, but further research would be needed to prove how dangerous it really is.
Okay, I did my science fair project on the human memory. Basically, the human memory is a system inside your brain that will store information that you learn over time. Just say, I have no clue what I'm talking about. Okay, we'll just start with my poster. Okay, we'll start with the poster. We'll start the poster, okay. The memory is a process of storing and retrieving information in the brain. A thing that helps some people is remembering the 4 R's of memory. Recollection, which is if you learn something, you can remember it again very easily. Recall, which is basically the same thing, you just remember it again. Recognition is if you see something, you can recognize it, which kind of refers to guideposts, which is seeing something that looks like something else. So you want to remember the 6, you look at the J, 7K, 8F, and 9P. That kind of helps you a little. And then relearning, if you've already learned something, then just kind of continually study it and learn it all again. Okay, down here are just some things I cut out of a magazine. This is the human brain, both sides, up and down. This is basically describing the structure of the brain and how it works. This is an article I cut out that tells how men and women's brains are different in shape and size and everything. This right here describes all the brain's areas, like where memory is stored, what controls the eyes and stuff. This picture right here is a picture about, say you're oversensitive about a part of your body, like your feet, then like if you were actually like, your hands, like this guy, if like you're actually oversensitive about it, they would actually be bigger because you care more about them than anything else. This is just the brain and the spinal cord and how some of the things work in the nervous system and send messages to other parts of the body. This is a sentence that my 8th grade teacher gave to us that tells about how you can remember the history of the United States in one sentence. And I don't have my test results on the poster, but I can tell you what they were. I was studying in memory for boys and girls. I took 5 girls and 5 boys, and I gave them 2 different tests. One was on the cards right here. I just gave them 10 seconds to study the cards and 20 seconds to get as many matches as they could. The highest amount of matches was 5, and 2 boys got 5 matches. The highest for girls was 4. And then my second test was I took a word association test. I got 2 words in 4 packs of words, so it was 8 words total. They were allowed, I read them out loud to them 3 times, and then I gave them 10 seconds to write down the second word in each sequence. And after I took care of all the results and everything, I found out that boys have a better memory than girls. Because I don't know why, it just sort of happened. Okay. What I was hoping to do, if I could have any volunteers, was to come up and try the first test with the cards. If I could have any volunteers that would like to try that. Okay, Grasso, come on up. And Renee, you can do it afterwards. Here, Nate, you just stand right there. I'll get to you in a moment. Okay, Pete, you can study the cards for 10 seconds. Okay. Here, Pete. You have 20 seconds to find as many matches as you can. Go. Stop. Okay, that's pretty. Here, Renee, study me 10 seconds. Okay. We're in routine. Okay, Renee, you have 20 seconds. Go. Yep. Stop. Eagles expert. Hey. I think Renee got more. What do you mean? I think she's cheating. You got 6. That's okay.